Dear friends, today we are going to discuss the topic phototropins, discovery, chemical nature, role in photomorphogenesis and the mode of action. The following objectives will be discussed during the present study to learn about the concept of light regulated plant development or photomorphogenesis. Number 2, to acquire the knowledge about the discovery of phototropins, to know about the chemical nature of phototropins, to gain an insight into the physiological role of phototropins, and last, to understand the mechanism of phototropin action. Plants utilize light not only as energy source for the process of photosynthesis, but also as an information source to enable the appraisal of environmental signals. They sense changes in the ambient light and adjust their development to optimize their fitness to the local environment. Plant cells carry several different classes of photoreceptors that can absorb different spectra of light. These photoreceptors include red for-red light receptor, that is phytochrome, and blue light photoreceptor, cryptochrome, and phototropins. Phototropins are blue light activated photoreceptors that play a dominant role in phototropism of plants. Phototropin protein contains two LOV domains, that is light, oxygen, and voltage domains in their N terminus that are characteristic of protein activated by light, oxygen, and voltage. Each LOV domain carries one flavin mononucleotide chromophore that absorbs light in the blue region of the spectrum. At the carboxy terminal end, designated as C terminus of the protein, is a serine threonine kinase domain. The photoexcitation of flavin molecule adds a cysteine residue to a serine threonine kinase domain in the C terminus of the protein backbone, thus activating autophosphorylation of the enzyme. The past decade has seen dramatic advances in our knowledge of plant photoreceptors and in our understanding of signal transduction pathway that they activate. A major part of these advances has been the identification and characterization of photoreceptors that respond to signals from blue region of the electromagnetic spectrum. One of the numerous physiological processes controlled by blue light is phototropism. Phototropism enables plants to bend towards incident light by perceiving the direction, wavelength, and intensity of incident light so that they are able to obtain maximum light. There are at least two classes of blue light photoreceptors. Number 1, the cryptochrome and the phototropins. Phototropins are blue light activated photoreceptors that play a dominant role in phototropism of plants. Cryptochromes regulate plant development and photomorphogenesis, whereas phototropins are primarily responsible for photomovement responses such as phototropism and chloroplast relocation. Phototropin was first identified as 120 kilodalton plasma membrane protein that undergoes blue light dependent photophosphorylation. The gene encoding phototropin was cloned from Arabidopsis mutant designated as NHP1, non-phototropic hypocotyl 1, that is impaired in blue light induced hypocotyl and the root curvature responses. The NPH1 gene or NHP1 mutant, NHP1 apoprotein and NHP1 holoprotein have been renamed as FOT1 and FOT1 respectively, designating phototropin and FOT mutant respectively. FOT2 is a phototropin 1 homologue in Arabidopsis that shares 60% sequence identity with phototropin first. Phototropin first and phototropin second contribute equally to stomatal opening, whereas avoidance of movement of chloroplasts is mediated by phototropin 2 under high light intensities. Now we will discuss the discovery of phototropins. Since 1930s, action spectra for phototropism have been repeatedly determined for a number of organisms, but have been most thoroughly documented for coleoptiles of oats and siamase, and for spongiophores of the fungus Phycomyces. The action spectra for oat coleoptiles and the phycomyces are virtually identical, indicating that they share a common homologous, if not common, photoreceptor. All other phototropic action spectra are similar and consistently show two peaks in blue region of the spectrum near 475 nanometer and 450 nanometer, and a small peak at 420 nanometer. In addition, there is a broad action peak in UVA region near 370 nanometer. The action spectra for both OAT and phycomyces phototropism shows an additional peak in the region of 280 nanometer, indicating that photoreceptor is probably chromoprotein. As early as 1940s, it was suggested that the photoreceptor could be a flavin molecule such as riboflavin. However, because of its action spectrum, the photoreceptor for phototropism was subject to the same flavin carotenoid controversy for other blue light responses, now known to be regulated by cryptochromes. On the other hand, there were several physiological results that ruled out carotenoids as the photoreceptor for phototropism long before the responsive pigment phototropin. It was finally discovered as shown to be a flavin protein. After the investigation of phototropin first and phototropin second mutants for blue light responses such as deethylation, photoperiodic flowering and stomatal opening, the monogenic mutant showed little defect in these responses. The lack of function of phototropin in these photomorphogenic responses, that is deethylation and photoperiodism, is not astonishing, as these responses are under the control of different kinds of blue light receptors called as cryptochromes. However, the stomatal opening induced under blue light turns out to be mediated redundantly by phototropin first and phototropin second. A study that compared stomatal opening in Arabidopsis mutans exposed to both blue and red light found that stomata in the phototropin first and phototropin second double mutants failed to open as compared to wild type plants. Under these conditions, because stomata open much wider in response to the combination of blue and red light than to red light alone, these investigators concluded that stomatal opening was a blue light mediated response controlled by phototropin first and phototropin second. Now about chemical nature of phototropins. Phototropins are blue light sensors that are responsible for range of responses including phototropism, chloroplast movement and stomatal opening that combine to increase the photosynthetic efficiency of plants. Initially identified in the model plant Arabidopsis thaliana, phototropins have since been characterized in the unicellular alga Chlamydomonas and pteridophytes and in several angiosperms. As with the phytochromes and cryptochromes, the primary amino acid structure of plant phototropins, phototropin first and phototropin second, can be thought to be as two distinct components: an N-terminal photosensory region and an accompanying C-terminal effector domain that includes a classic serine threonine kinase motif. The N-terminal photosensory region of phototropins contain two photochemically active LOV domains designated as LOV domain 1 and LOV domain 2. Each of these domains bind to the blue light absorbing cofactor, that is flavin mononucleotide designated as FMN, in a non-covalent fashion as chromophore in the dark. The combination of these domains allow blue light dependent phototropin autophosphorylation in the vivo and in vitro conditions. LOV domains have a highly conserved tertiary structure which facilitates the non-covalent binding of FMN or FAD. Phototropin and other plant LOV domains incorporate FMN, whilst fungal LOV domain contains an 11 amino acid insertion to accommodate the larger chromophore, that is FAD. The structure of LOV1 has been crystallized from the species, that is Chlamydomonas phototropin. whilst the structure of LOV2 domain from adiantum. The structures of LOV1 and LOV2 are almost identical and are likely to be good models of love domain structure given the highly conserved primary structure of the motif. Love domains share a common structure consisting of 3 helical segments and a 5-stranded anti-parallel beta barrel which forms a hydrophobic pocket in which the flavin chromophore resides. Autophosphorylation of phototropin plays a significant role in the phototropic responses, probably by initiating a phosphorylation cascade. Studies employing mutants of FOT1 gene have shown that the protein is apparently folded in such a way that phosphorylation site is blocked by LOV domain 2 in the dark. Absorption of blue light by chromophore induces a change in the conformation of the protein so that phosphorylation site is available and becomes active. The role of LOV domain 1 is not clear. Mutants lacking the LOV1 domain have shown that LOV1 is not necessary for phosphorylation, but its presence does increase kinase activity of the protein. Now we will discuss role of phototropins in photomorphogenesis and its mode of action. Plant phototropism, the ability to bend towards or away from light, is predominantly controlled by blue light photoreceptors, the phototropins. Phototropins have been found in various plant species. The structural properties of phototropins are highly conserved from the unicellular alga Chlamydomonas to higher plants. To date, phototropins have been identified in several plant species and are ubiquitous in higher plants. The phototropins of Chlamydomonas regulates the sexual life cycle in response to blue light. Regardless of their distant relationships, Chlamydomonas phototropin is functional in both Arabidopsis thaliana and Chlamydomonas, suggesting that the basic mechanism of action of phototropin is highly conserved. There are 2 phototropin homologs in Arabidopsis designated as FOT1 and FOT2, which share approximately 60% sequence identity. FOT1 and FOT2 have overlapping and distinct function in Arabidopsis. They function redundantly in phototropism, the chloroplast accumulation response, stomatal opening, and leaf flattening. However, phototropin 1 is much more sensitive than phototropin 2 in some of these responses. Although both phototropin 1 and phototropin 2 have mediate chloroplast accumulation response, the avoidance response is solely mediated by phototropin 2. In addition, phototropin 2 but not phototropin 1 is required for chloroplast accumulation at the bottom of cell in darkness, known as dark positioning. The functional specificities of phototropin 1 and phototropin 2 could be attributed to differences in the gene and protein structure between phototropin 1 and phototropin 2. However, little is known about the structural basis for the functional specialization of phototropins. The role of FOTs in initiating the variety of responses is discussed below. Phototropism. The phototropic responses may be separated into 2 distinct stages. First, positive phototropic curvature occurs under very low flux and obeys Benson-Roski law of reciprocity, that is, the same degree of curvature is obtained for a given quantity of light, regardless of the flux time combination used to supply the light. This initial response differs from second positive curvature that occurs in a time-dependent manner in response to long-term exposure to light and results in large organ curvatures which are apparent to the naked eye. The FOT1 locus was initially identified in a screen for mutant Arabidopsis seedlings defective for hypocotyl phototropism, and as such this physiological response provided inspiration when FOT nomenclature was rationalized. In Arabidopsis, both FOT1 and FOT2 have a role in mediating this response of phototropism, with FOT1 conferring sensitivity at lower light intensities and phototropin 2 being functional at higher fluence. Such differing roles are partially explained by differential gene expression. While only phototropin 1 is expressed in etiolated seedlings, whilst phototropin 2 transcription increases upon exposure to light. The shorter half-life of phototropin 2 photocycle reversion may also allow phototropin 2 to retain sensitivity at higher fluences. PHOT activation has been biochemically linked to auxin redistribution via a phototropin 1 interacting partner. Non-phototropic hypocotyl 3 NPH3 is necessary for phototropism in Arabidopsis and interacts with the photosensory N-terminal of phototropin 1. NPH3 interacts with phototropin 1 in vitro and undergoes dephosphorylation upon blue light exposure. Phototropic curvature is mediated by an increase in growth on the shaded side of the stem resulting from an accumulation of plant growth hormone auxin. At present, little is known about how phototropin activation by blue light leads to accumulation of auxin on the shaded side of the stem. Unilateral irradiation has been shown to induce a gradient of phototropin 1 autophosphorylation across oat coleoptiles, with the highest level of phosphorylation occurring on the irradiated side. A phosphorylation gradient model has therefore been proposed to account for the complex fluence response curve for phototropism. But the question still remains as how a gradient of phototropin phosphorylation across the stem can bring about the lateral gradient in auxin. Phototropin controlled chloroplast movement. Chloroplasts are distributed in low light in a manner that presumably maximizes light capture for photosynthesis. By contrast, they are redistributed in high light in a manner that minimizes light interception, presumably to reduce photo damage, called as avoidance response. As with phototropism, FOT1 and FOT2 exhibit partially overlapping functions for light-induced chloroplast migration. Two research groups have independently described the impaired chloroplast migration responses for FOT2 single mutants of Arabidopsis. Each study showed that the avoidance response is absent and that the accumulation response can occur even at highest light levels so far tested. Evidently, FOT2 activated at high fluence rates counteracts the action of another photoreceptor that mediates the accumulation response. Both studies also found that FOT2 gene expression is upregulated by light, indicating that FOT2 probably predominates under high light conditions. It has been reported that although the high light avoidance response is normal in the phototropin 1 mutant, the accumulation response shows a somewhat higher light threshold requirement than in the wild type. This result indicates that phototropin 1 somehow contributes to the accumulation response, but is not the only player. It was only when chloroplast migration in a phototropin 1 or phototropin 2 double mutant was investigated that the situation was clarified. In this mutant, chloroplasts show neither an accumulation nor an avoidance response, regardless of the light treatment given to the plants. Hence, both phototropin 1 and phototropin 2 can mediate accumulation response of chloroplasts to low intensity blue light, although phototropin 2 is somewhat less sensitive than phototropin 1. Only phototropin 2 mediates the avoidance response and it counteracts phototropin 1 action under high light conditions. Two proteins have been identified which are required for both chloroplast accumulation and avoidance response. Chloroplast unusual positioning 1 designated as CHUP1 and plastid movement impaired 1, that is PMI1. CHUP1 is an F-actin binding protein which is consistent with the known requirement of cytoskeletal rearrangements for chloroplast movement. And the N-terminal region of CHUP1 is sufficient to target green fluorescent protein, that is GFP, to the chloroplast envelope, suggesting that CHUP1 may mediate interactions between the chloroplast and the cytoskeleton. PMI1 mutants also display defects in both chloroplast accumulation and avoidance movements of chloroplasts despite the cytoskeletal structure of PMI1 mutants appearing to be intact. PMI1 is a protein with unknown function, although it has homology to several signaling peptides. Now we will discuss blue light mediated stomatal opening mediated by phototropins. The opening and closing of stomatal pore is controlled by the movement of a pair of guard cells which surround each pore. Because of the vital role of the stomata in regulating gaseous exchange between leaves and the environment, stomatal opening is tightly regulated by several internal and external mechanisms including circadian cues, intracellular carbon dioxide levels, drought, and both red and blue light. Blue light dependent stomatal opening is controlled by both cryptochromes and phototropins. The data suggests that phototropin mediated stomatal opening occurs at higher light intensities than those required for cryptochrome activity. Blue light has been shown to rapidly induce stomatal opening in a phototropin dependent fashion as the Arabidopsis FOT1 and FOT2 double mutant demonstrates an impaired response, while blue light specific stomatal opening was restored completely by the wild type construct. Stomatal guard cells of the FOT1 and FOT2 double mutant fail to extrude protons in response to blue light treatment. Proton extrusion is essential for stomatal opening and involves the activation of the plasma membrane H-positive ATPase. Activation of guard cell proton ATPase involves phosphorylation of the proton ATPase and 14-33 binding. 14-33 protein belongs to a highly conserved protein family that typically bind to phosphorylated target proteins and regulate signaling in eukaryotic cells. Similarly, FOT1 from Vassia faba broadbean root tips and guard cells binds to a 14-33 protein upon autophosphorylation. Specifically, 14-33 binding to Vassia FOT1 requires phosphorylation of serine 358 situated between LOV domain 1 and LOV domain 2, which is equivalent to serine 325 of OAT FOT1, that is phosphorylated in response to intermediate fluences of blue light. Thus, one of the consequences of phototropin autophosphorylation is to mediate 14-33 binding. One might speculate that the binding of 14-33 protein may facilitate direct interaction between phototropins and the guard cell proton ATPase pump. Yet, the fungal toxin fucoxin has been shown to induce phosphorylation of proton ATPase and subsequent 14-33 binding protein in absence of FOT1 and FOT2, implying that some other protein kinase is responsible for phosphorylation of the proton ATPase. Indeed, the signaling pathway between the phototropins and the proton ATPase was recently reported to involve the activity of type 1 protein phosphatase in broad bean god cells. Further work is now needed to clarify the functional significance of 1433 binding protein, especially since 1433 binding protein to the phototropin has also been observed in etiolated seedlings. Now, calcium dipositive as a potential intermediary of PHOT signaling cascade. Phototropin activation leads to increase in cytosolic calcium dipositive concentrations. Calcium dipositive has previously been shown to be a versatile intracellular messenger, and changes in intracellular calcium levels are observed in stomatal opening, chloroplast movement, and the rapid inhibition of hypocotyl elongation, which are all blue light responses initiated by phototropins. Both phototropin 1 and phototropin 2 are capable of increasing cytoplasmic levels of calcium dipositive, although the calcium dipositive source used varies from species to species. The correlation between intracellular calcium and phototropin mediated signaling to light dependency, as both phototropin 2 mediated calcium dipositive release and phototropin 2 mediated physiological responses require higher blue light fluorescence rates than those depend on phototropin 1. Interestingly, it has been speculated that phototropin 1 may suppress phototropin 2 mediated calcium 2 positive import in mature leaves. The blue light and ultraviolet A light regulate a wide range of responses in plants, including phototropism, chloroplast migration, and stomatal opening. However, the photoreceptors for these light responses have been identified only recently. The phototropins, that is PHOT1 and PHOT2, represent a new class of receptor kinases that appear to be exclusive to plants. Recent genetic analysis have shown that phototropin 1 and phototropin 2 exhibit partially overlapping functions in mediating phototropism, chloroplast migration, and stomatal opening in Arabidopsis thaliana. Although significant progress has been made in understanding the early photochemical and biochemical events that follow phototropin excitation, the details of how this excitation activates such different responses remain to be elucidated. Thanks for watching.